So we talked about what field work is. Um, so an important part of this is measuring populations. And the techniques differ a lot depending on what type of organism you're studying. So these show some examples. So this elk has a radio collar or satellite collar on, and that will track movements of this individual. And if it's a radio collar, somebody needs to go out with an antenna and locate the individual every time they want a location for the individual. If it's a satellite transmitter, the data will be uploaded automatically, and all you have to do is get on your computer and you can get information about where that individual was various points during the day. So I've worked with data on like this for lesser prairie chickens, which is like a ground nesting grouse type bird. And we had ours to set to record the location every hour. And it depends on the battery life of your satellite transmitter. But basically, you can upload from satellites all the information about where your individual was over different periods of time. This is a similar thing. They make backpack ones that large birds can carry. I've been involved with putting them on swans and ducks, and you can see this raptor is very large, and so it can handle one of these. Little songbirds usually can't carry these. This person is studying corals, and so they are measuring them over time to look at growth rates. And you can see the equipment's very different if you're working underwater. This is a quadrat where people are documenting the number of different species of plants. So methods vary a lot depending on what type of organism you're working on. So there are a couple of ways to look at population dynamics or numbers of individuals in a population through time. A census is where you count all the individuals in a population. So for species that are easy to detect, these are easy and cheap. So you go out, you count everyone you see. Like for trees, for example, it's really easy to detect a tree because they're big. They don't move. You can tag it and see how big it is, and then you can go back the next year and count all the trees. Repeated censuses can reveal change in population size over time. So depending on what type of organism you're looking at, like a tree, you could go and probably count them every 5 or 10 years. You wouldn't have to go every year. For smaller things, you might want to do more repeated censuses. So it depends on the time span for that species. So this is an example of census data on where they went out and they counted all the individuals of Hawaiian monk seals. And you can see you get time data like this where you have the number of seals over each year, and it gives you the different range of data. And you need this kind of data before something's declared endangered, because otherwise you don't know what the population size was before. So it's hard to know if a species is declining if you don't have any information on how many of them there are. Here's another example from orca sightings. They can identify orcas by the marks on their tails, and they can count up the individuals. And so you can see here, this looks at the number of them in some different bays, different areas in the Hudson Bay region. And this indicates that they're increasing in abundance in Hudson Bay. So you can see that they're counting more individuals. And this is by decade. So a census is going to count all the individuals in a population. And this is really difficult for a lot of species. If they're secretive or they live across a really large area, you can't count all the individuals in the populations. Secretive makes it especially hard. So I study some salamanders that go underground. You can't go out and count all the salamanders. It's pretty much impossible. You can count a lot of them as they come in to the pond to breed, but even that's not perfect. It's not really a census. We can't be sure that we got every single animal. So a lot of times in this case we do surveys. And you count a sample and use that to estimate the total population size. And so these are some examples. You can count the number of partridge pea plants in 30 random 10 by 10 meter quadrats. A quadrat is just this square thing that you put down and you count all the plants in it. And you pick random locations to put these quadrats down, and you count all the individuals of a particular species, and you might do that year after year to get an index of the population size. You don't know exactly how many individuals there are in a whole field, but you can get a decent estimate. You might also do a mark recapture study. This is a mist net setup to catch songbirds, and you can put bands on them and estimate how many new individuals you're finding, and from that you can extrapolate an estimate of the population size. So some more examples. It again really depends on what type of organism you're looking for. So you might again use these quadrats, which are usually square frames that you put down, and you count all the individuals inside of that. You might look at the number of individuals you're catching in some sort of net or trap. You might use a transect, which you can see it illustrated here, which is where you put down a long line, and you might either use a transect or look at all the individuals that are within a certain distance of this transect as a way to sample the environment. You might count the number of individuals calling. A lot of times for waterfowl, they do aerial surveys. So the biologist goes up in a tiny plane and they fly over areas that look like this. And the biologist has to estimate the number of individuals in this large group. And obviously it has to be on days where they're easily distinguished from the vegetation they're in. So I believe these are snow geese, and each little white dot is a goose. And so you can imagine on a day where it snowed, it wouldn't work because they would blend right in and you wouldn't be able to tell the difference. You can also put up things like camera traps where movement triggers the camera to take a picture, and you can sort through those and look at the number of individuals that you took a photograph, or just the number of times you see an individual. Sometimes you can't tell them apart, so you can't necessarily say that it's the same individual that's walked by a few times or several different individuals. You can also get counts from signs of animals. So you can do counts of hair left behind or tracks, so footprints, or scat, which is poop basically. And you can identify animals by their tracks and by the shape of their poop or their scat. And by doing those kinds of counts, you can also get an index of how many individuals there might be in an area. A really robust way of doing it is mark recapture, where you catch a sample of individuals with traps or otherwise. You put some sort of mark on them, and then you go out another time and you look at how many individuals you get that aren't marked and that are marked, and from that you can extrapolate the population size. And that's relatively robust, and there's a lot of science and math behind that sort of methods. So a survey looks at populations over time with a standardized protocol is particularly valuable. So you need to use the same method through time, and doing that over a long period of time can get you a really good idea of what population fluctuations look like, especially in these species where it's impossible to count every individual. If you use a standardized protocol, it ensures that the effort is the same over time. So you don't want to go out one year and spend 40 days trapping for an animal, and then the next year spend 2 days. You're not going to be able to compare the counts across time. But if you standardize exactly what you do, you can make these robust comparisons. And as I said before, a lot of times you don't even need to see the organism. A lot of tigers are surveyed using paw prints, bears using their scat, and lately people have even been using DNA. And so for aquatic organisms, you can take a water sample and run PCR and look at their genetics, and you can get an estimate of how many individuals or whether that species is present in a pond or stream. So this is some data looking at how well they call it eDNA, environmental DNA, and that's sampling the environment for DNA. And they've done looking at how likely you are to detect different species, knowing that they're already in that water. So basically, they did a sample and trapped them, and they were sure that that species was in the water, and then they took water samples looking for eDNA to verify whether this technique was going to work for different species. So they find relatively high detection in ponds where the water stays in one place. And you can see it's 100% for common spadefoots and for tadpole shrimp and this loach fish, and it's 90% for this species of newt and 80% for this dragonfly. So pretty high probability of detection. However, in streams, it's much lower. And so they had a much harder time detecting Eurasian otters with DNA, but a reasonable success rate with this loach fish in streams. So this also looks at salamander DNA. And you can see that they looked at how much DNA was shed in a salamander in the laboratory over time. And you can see that if they had been in the water for about a day, 24 hours, then they could detect that DNA here, you can see. But once the salamander left, the DNA signal went away pretty quickly. So it depends on how many individuals there are and in how much water, how long they're there, whether they're actually present when you take the water sample, and it doesn't last incredibly long. But people are working really hard on this method. So there's different disadvantages of surveys and censuses. We talked about some of the advantages. Some of these can be very time consuming. Sometimes the results are very location specific. So just because you know there are this many individuals in one area doesn't mean that it necessarily translates to another area. A lot of times these censuses and surveys are discontinued after a period of time due to lack of people and financial support. And many species are really difficult to detect or could be negatively impacted by the censusing effort. And sometimes we develop new survey methods, or some are just too time consuming and we have to switch survey methods. And that makes it difficult to compare across time.